Hello and welcome back, friends. We have just discussed about the basics of FASTA and BLAST algorithm and what are the type of algorithm and why we are using FASTA and BLAST algorithm and what is the importance of this type of dynamic programming because they are advantages over the pairwise alignments because this type of programming are faster than the pairwise alignment. Now let's talk about FASTA in this video. So FASTA, it is derived from a logic of dot plot actually. So we need to understand what dot plot is before going into the understanding of FASTA algorithm. So it is a modified version of dot plot and implication of dot plot, which is a compute based diagonals for all the frames of alignment. We'll be seeing it later. Now the method looks for exact matches between the words in query and the test sequence. Now wherever we are making alignment, we are having two sequence in our hand. One is our query sequence, which is a sequence of our interest. Another one is the test sequence, which we get from a database. So query sequence and test sequence are aligned together. How can we align them? The actual formula here to match the exact words. So exact word match is important for this case. Now the exact word range that in case of FASTA is taken for the match is for DNA it is 6 nucleotide words and for protein it is 2 amino acid long words. Now if you look at the FASTA form algorithm outline, what we get, these are 5 steps to get the FASTA algorithm. One is to identify the common words. These are called the K words, but these are simply the words. For example, if it is a nucleotide, then A, G, T or C, these things can provide the words. Say A, G is containing the range of the word. Now we have seen here for nucleotides the word range can be up to 6 nucleotide long, so it can be AGTCCA for example. So say AGTCCA, this whole thing can act as one keyword. Okay, so this is what we call keywords. 6 for nucleotides and 2 for proteins. Okay, now so identification of these common keywords between I and J. Now what do you mean by this I and J? One of this I is called a query sequence, one of them is called the test sequence. For example, this query sequence is designated as I, test sequence is designated as J. Now the second point is after identifying the exact matches between this query and test sequence, scoring of the diagonals with keyword match. So we'll score them depending upon the match and scoring the diagonals. Here comes the important part. We have seen here the scoring of diagonals. So what we mean by the scoring of diagonals in this case? Scoring of diagonal means we need to understand the dot plot because in the dot plot we create diagonals. Now I'm going to tell you what diagonals are in a moment, but just understand this when we create a dot in this way, so we just join the dots to make these diagonals in the matrix. Now we need to look for the scoring of these diagonals to get the best diagonal scores. Okay, now the third part is the rescoring of the initial region with a substitution score matrix. So we'll change the element of score, we change the parameter of the scoring using a different substitution scoring matrix. Fourth part is the joining of initial region using gaps, and then finally perform the dynamic programming to find the alignments. I know these things is difficult for you to understand, so let's go back, let's come to the discussion in detail. So first part is identifying of common keywords between I and J. Remember this one, identification of common keywords between I and J. So here it is, the word matches in two sequence. Suppose so this is the matrix for ours. So this is one coordinate, this is another coordinate, and this is from the beginning, for example. So what we get in one part we add, so in this case the coordinates are in this direction, sorry for, so the coordinates will be, so let me erase this ink, sorry. So let me take another color. Okay, now in this case suppose this one, so these are this green color region are the example of coordinate. Now here in this coordinate we put j values or a sequence, which is in this case the test sequence for example, and in this case this is suppose the query sequence. We put it in this coordinate and we look for the match between this j value and I value. So we need to look for the matches, right? Now say here the I sequence is A T. So suppose the sequence is, say here it is A, then it is T, then it is C, then it is G, then it is G, and so on. Now in this J part we are having T, G, G, T and G and T and so on. So when you go and look for the exact match, when you can find, in this case we find this, say here it is a G and here it another G, so this two G match in this point, so create a dot in this matching region. Okay, similarly if this T matches with this T, we create him a dot here, so we create dots depending upon the match from both the coordinates to get our dots in the matrix. Okay, so this is a simple construction of dot matrix depending upon the keywords. Now remember, I have discussed to you about the keywords. Keywords for nucleotide is 6 sequence long, it's for protein 2 amino acids long. Now here dot matrix elements are called this I and J. One is query sequence, uh, another one is the test sequence. Now here what we get, if the word starting at the position I in this I is identical to the word in position J, we create a dot at that position. So this is the basic rule. Now when you get this kind of dots ready for the next round of all is scoring diagonals with keyword matches. Okay, so now as we get this word match, so we can say different. So this is a dot matrix. In this dot matrix we can see this is a J and this is I. Okay, now it is the query sequence and this is the test sequence, for example. Now in this I we are having G C A T, this is the sequence in this direction, this is the sequence in this horizontal direction. Now say C and G no match, so no dot. C C a match, so one dot here. A is a match, so a dot here. T with this T matches here, so a dot. But T with A no match, so no dot. T with C no match, no dot. So here again C with C, here it is a match. C with C, here it is another match, and we'll be putting these matches, so we'll be creating dots in this place, this place, this place, sorry, not in this place, not in this place, this two C matches. Now this C again matches with this C, so create another dot. Now this G again matches in this place, so we'll put another dot here. This G matches here, another dot. This G matches here, so another dot here. So we'll be putting dots all over this dot matrix to get our scattered dot score. But after taking the scores, we need to score the diagonals with the keyword match. And after scoring the diagonals with keyword match, we'll creating the diagonal. For example, say in this case we have provided many scores or many dots, right? So throughout the place we created dots. Says this is a dot for C, this is another dot for this C, dot for C should be there, another dot here. So there should be many dots in this matrix. But you can see here that we have only joined those dots which are creating a diagonal line. Remember, so this is what we call a diagonal during this dot matrix plot, because we not only plot the dots but we can only join those dots which are forming the diagonal lines. Now, in this case the diagonal line forming dots are joined only, but rest of the dots, say this one, this red dots that I have put, these dots are not creating any diagonal lines, so we cannot join them, because this is our starting point, because this is our zeroth point, and from here we are going this two direction coordinates, so we must create the diagonals depending upon this direction. So that's why we create or take only these two diagonals by joining the dots but not joining other dots. Okay, so this is very important. Now the third part is rescoring the initial region with substitution matrix. Now you can see here, once we get this kind of diagonal lines in a dot matrix, so suppose here a lot of different small and long diagonal lines are created due to the dot matrix joining with diagonals. After that, what we need to do is to uh, look for the scores. Now remember, we have made the scores and the score is given by I minus j. This is the score value that is given. Okay, so after giving this score value, what we get, for example, say in this case, in the first case, so 1 2 3 4 5 6, so the j value here is 6 and this I value is 1, so 1 2 3 4 5 6 and 1, so the I value is 1, so what we get, the value is -6. The diagonal of this one is having the value -6. Okay, so that's how we score the matrix. After scoring the matrix, this is not also that much important, so I'm not focusing on to that. But anyways, now after giving the score onto the matrix, we need to use a rescoring of that, because the score that we provide depending upon the I and j matrix values are not sufficient for getting the desired result. So we need to use some other scoring matrix, which is a best scoring matrix. For example, here we utilize the PAM matrix as uh, the rescoring matrix to rescore our alignment, because in this case what we use simply dot matrix alignment, which we can rely on. So we use another matrix, which is PAM matrix, for rescoring them. We rescore this using PAM matrix, and after rescoring we get this desired result. Now why, remember, in this case of simply I and j dot matrix scoring, we get the depth of these lines, or the bold nature of the line, is telling us how much better score they get. And we can see almost all of these diagonals are having same scores, because they are having same depth. That means they are having same scores, right? But this is not the case, because all of these diagonals are not good alignments, so we cannot give all of them the same score. That's why we need to rescore it with a better scoring matrix, which is PAM matrix. Now after scoring them with PAM matrix, what we get? Differential scoring values. Now this value is less than this one, because you can see this is pretty much dark and bold. That means it is telling us this is having a higher score in PAM matrix than this one, because these things are very lower scoring regions, these are higher scoring regions. So what we do now, after rescoring them using PAM matrix, we are enabled to take up the best scoring results, or the best scoring diagonals. So for the for providing, or for taking the best scoring diagonals, which are also called inteen or init. Now, init means those scoring diagonals which are best match. Okay, so here these darker ones are called the init. Okay, now joining of the initial regions with gaps. Now what we'll do, we'll take only those which are having the dark regions, or which are having the higher scoring matrix. So scoring matrix value. Now here said these are the darker ones, so we'll take the darker one only. So you can see we discard all the rest of the scoring matrix values, the light color values. We only take those one which are having the higher scores given by the PAM matrix scoring matrix. Okay, now as we get these values, what we'll do is simply join these regions with gaps. Okay, simply join them with gaps. So let us start joining with gaps. So how can we join them? Simply by like this one, then join this one, then this one. Now as we are joining them using gaps, gaps are not good. Remember, I have always told you that gaps are not good, they are bad, because gap mean there can be any different type of sequence can be present there. So suppose a gap in nucleotide, that means any kind of sequence, any kind of base can be put there, like A, G, T or C, so on. If a gap in protein sequence, there can be any of the protein amino acid among 20 amino acid sequences, so they are not good. So we must penalize this kind of alignment for the gaps. So we deduct some scoring value by adding these gaps, and then finally get the optimum scoring value. For example, it is previously getting a scoring value, and adding all the scoring values for the diagonals, we get a total scoring value T. Now by adding these gaps, there are some negative scoring values for gaps, so we'll extract or subtract this negative scoring value from this total scoring value that was previously there. Now we get our desired scoring value in this case. Now we'll compare these desired scoring values for several different alignments that we have done using this matrix, because this is not only one alignment we have done using FASTA. FASTA analysis is done for many sequences, so we get many of this kind of final scoring value by adding this gap and providing the penalty. So we get all of this D1, D2, D3, all of these desired values, and we'll compare which is the best scoring value. We'll take, suppose D2 is the best scoring value, we'll take it and take that alignment which is the response of D2. So we'll take that alignment, and we will be using that alignment as a ideal one. Okay, now what we can do is this use of the last part is use of dynamic programming to create the optimum alignment. Then use the dynamic programming method to align those sequenced optimally, which from which we get the desired best value. Okay, so this is a result of a first alignment, and among all what are important thing is this init. We have already talked about that. Z score is important, as well as the E value is important. Now what is Z score and E value? Let us look at in the future slide. Now in this case we can see the alignments are shown in this particular place. Now we not only have to align the sequence, but after each of the alignment, whether in FASTA or BLAST, we need to assess our alignment, whether the alignment we have done is correct or not, and how much it is correct or not. Utilizing, we need to look it utilizing biostatistics or statistical analysis. Now what we can do here, generation of the random alignments, we can calculate. Then we need to compute the mean and the standard deviation for the random scores. Remember, we provide the random scores, so you need to look for the mean and the standard deviation for the random scores. And also we need to compute the deviation of the actual score from the mean of random scores. So we need to look for what are the type of deviation, what is the amount of deviation that is done uh, from deviation of the actual score from the mean of a random score. So what is the actual deviation? So we get this value if we divide the mean of this random score by the standard deviation, and that is called the z value or z score. Now the z score or z value means simply the deviation of actual score from the mean of random score. So please try to think about this concept, that z score means how much deviated our actual score is from the our random score is from the actual score. Okay, so how much sorry, not how much z value means how much our actual score is deviated from the random score, right? So we are giving the random scores, and the random score we've got in all these cases, they are randomly placed. So we take a mean of all the random score and divide it with the standard deviation, so that we get an optimum score value. Now this is telling us about the deviation. Now as this value is less, so we get a less deviation, a better result for our. And also what is the e value? Now the probability of z, or the z value, is called the e value. Simply the probability of the z value is called the e value. So if the probability of the standard deviation, probability of the deviation of the actual score from the random score is high, that means our assessment is not that good, our alignment is not that good, because it is varying the actual score from the mean of random scores. So that means higher z value means so that means a higher deviation, or maximum deviation, or better deviation, and more deviation means it is less effective. So the probability of z value is called the e score. So less the e score, so suppose what it means, then deviation, if the deviation is high, z value will be high, e value will be high. So, which is not good, because it is creating, or it is indicating us much more deviation of the actual score from the random score. That means we require less deviation. That means for the less deviation we have less z value, we have less e value, right? So we want less e value. Less e value is meaning, is telling us that better alignment is done. So we're always looking for less e values and less z score, because it is indicating less deviation of the actual score from the mean of random score. Now here the rule of thumbs for e-values. Now e-values below 10 to the power minus 6 are most probably statistically significant, and they are good. E-values above 10 to the power minus 6 but below 10 to the power minus 6 can be taken and can be look for a second look. And e-values below e-value above 10 to the power minus 3, which are much more higher, should not be tossed aside lightly. They should be thrown out with a great force. So we cannot rely on the e-values, or we cannot rely on the alignments which are telling us the e-value greater than 10 to the power minus 3. We cannot take them, we should not take them. We should take only the e-values which are below 10 to the power minus 6 most of the time, because that is the most optimum one. Now this is as we have discussed about the FASTA alignment and FASTA algorithm, but there is a FASTA format also. This is a most reliable and suitable format that we can get in most of the softwares online nowadays available. You can see there in NCBI, when you take out nucleotide sequences or protein sequences, this is called FASTA format. Now in this format, it is used by almost all programs nowadays. Starts with this kind of header. So you can see here, this is the start with header, and then it will have some description. Then simply it will add the sequence in capital letters. So no specific requirements for line length, or characters or anything like that. So simply just letters randomly placed one after another. So sequence letters are placed in capital letters. So simply this is the FASTA format. Okay, so that's it, and I hope this video is helping you. Thank you.